Hi everyone, my name is Renzo Bonifazi. I am a researcher at Wagner Livestock Research. This is a work that we did in collaboration with University of Alberta within the BovReg project, which is part of EuroFAANG. So in this presentation I'm going to talk about biology-driven genomic predictions for dry matter intake within and across breeds using whole genome sequence data. The crossbreed is not yet done, so I will present on the within breed. So first of all, I would like to acknowledge all the people that collaborated in this work, which you see listed here. So we have heard about biology-driven genomic predictions, and well, I will go fast here on the introduction, but in general, to make usage of these millions of variants from whole genome sequence, we really need to pinpoint the causal variants across different traits of interest. This can be important for traits that have some biological effect that is complex, like biological efficiency of the animal, such as feed efficiency. So in BovReg, we have this catalog of functionally active genomic features, which I believe is what we so far call functional annotations. And so we can use this multi-omics data at genomics, transcriptomics, epigenomics, and chromatin accessibility for genomic predictions. And our approach has been to prioritize the SNP based on these functional genomic features and use them in so-called biology-driven genomic prediction, with the expectation that they could help for traits that are usually with small reference populations and that are related to efficiency, such as feed efficiency. So the aim of this study was to validate these biology-driven genomic predictions using genomic features on dry matter intake, and we did that using data from two countries, the Netherlands and Canada. From the Netherlands, we had data on Holstein, and from Canada, data was on beef cattle, and these were crosses with quite structured, quite an unstructured population. Here you can also see the number of herds, and as dependent variables from the Netherlands, we had the regress proofs for DMI, and for the Canada was pre-corrected DMI for fixed effects and the measured breeds. As number of animals in the Netherlands, we had 3,000 animals with genotypes and pseudophenotype, and then 5,500 animals from Canada. So we set up genomic prediction, and we use a forward-in-time validation approach where you basically define a year of birth, and all the young animals fall into your validation set, and you train on the older animals, and we obtain standard errors using bootstrapping. And here you can also see the number of the split, but in general we have about 25% of the animals in the validation. Then we imputed from 50K up to whole genome sequence using Beagle, and we proceed to select extra SNPs and prioritize variants based on genomic features. These are the genomic features we used from previous work packages. We have more, but we focus on these ones. We have QTL, EQTL, and ATAC-seq. And here you can see also where they were collected. Here I'm not going through all the list, but for QTL we have production traits, but we also have feed efficiency, methane, so more novel traits. For EQTL, we have tissues related directly to the trait itself, so like milk or mammary gland or the muscle, but we also have rumen and liver. And for ATAC-seq, this comes from the presentation that Gabriel will present after, I believe. He will give you all the details of how it was collected, but in general were 2 time points on 6 animals and different tissues as well. Then if you are like me and you don't know what is ATAC-seq, well, I include a slide. Basically, it's about trying to understand what part of the DNA is accessible. And so what this figure illustrates is that DNA is quite wrapped around around histones, and so it can be inactive and inaccessible, or at least not directly accessible. And the part that is open is more active. So with wet lab, basically, we are able to cut these parts that are open and amplify and sequence them and have a look into these parts, into this sequence of DNA, and understand which part of the genome is actually more accessible. And the idea behind is that here we may harbor regions that are promoters, enhancers, or regulatory elements. So what Gabriel did, and he will give you more details, I believe, in the next presentation. Across all samples, you get these peaks, and each peak is related to an animal sample, and what he did, he created for us consensus peaks, basically taking the interval that encompass, so to say, all the other intervals. So big part of the work was on processing all this data from different work packages. This is in short, the pipeline that we developed. The colleague from Alberta University created a big VCF files where basically you have all the genomic features mapped to the 1,000 whole genome sequence project, bull project, for all the traits and tissues. And here they store the position and the intervals and the p-values. Then we could iteratively select a genomic focus, functional group, such as QTL or EQTL, and here we just go for all of them. Our approach was quite direct. We didn't choose specific tissues, we use everything. And we extract all the variants that overlap under this 1,000 bull project. Then we filter out those that were already in the 50K panel and those that were not within our whole genome sequence, either in Canada or the Netherlands. And we use the p-values for LD pruning, so we do clumping. Then with this variance list, we can extract the biallelic variants, and we are basically down to having SNPs as a genomic feature layer for genomic prediction. So these are the scenarios we put in place. We have the 50K scenario. This is our reference one, where basically you can see in the table the number of SNPs per each country. We use the NextGP Julia package from Emre. And here you can do different type of Bayesian modeling. And for the base 50K, we decided to do what is maybe the most common. So a SNP-BLAP approach internally is bias seed 0. But the main assumption here is that you have common variance across all SNPs. Then we fit each genomic feature as an extra layer separately, either as QTL or EQTL. And you can see again colored the number of SNPs added per each layer. And for these layers, we use either SNP-BLAP or a Bayesian two-mixture model. But yeah, I will give away a bit of results. We haven't seen much difference on this Bayesian approach, so what I'm going to present is for SNP-BLAP. Finally, we combine everything, so all the layers together, and also include the information on attack-sec, so knowing if the QTL or the EQTL is in a accessible or non-accessible area in the DNA. So this, I will call it multi-genomic features, and as you can see, the numbers don't sum up. That's because we have overlapping genomic features, and we need to model that. So perhaps you have already followed the presentation yesterday, but when you have multiple genomic features with layers that overlap, well, how it works internally in the software is that you have different variants or different SNPs, and then you have to tell to the software, well, for example, SNP1 is present in annotation 1 and 3, and SNP2 in all of them, and so on and so far. So how does that look like for the Netherlands? Across the 17,800 variants that we had, we have 21 variants that are overlapping between QTL and EQTL, and 16 of them were into attack-sec regions. Then for QTL, you see that about 50-50 are in attack-sec and 50% are not, and 60% of the EQTL were in attack-sec and the rest not. So these are all information that has passed into the system, and we use a bias RCPI approach, a variation of it, where instead of the 4 classes proposed in the paper of Mollandin, we use 2 classes. So the software uses this information to allocate the SNPs to the class that explain more variance or to the class that don't explain much variance. So into the results, here you see all the scenarios and the prediction accuracy for the Netherlands. The one in gray is the 50K, is the scenario that we have to beat, so to say. And then you have each annotation layer on top of the 50K, and also we fit QTL and EQTL alone separately just to see what happened. And what you have here in the green is and in the boxes is the relative increase in accuracy relative to the 50K. So as you can already glance by eye, there is no much to gain in adding QTL and EQTL. We haven't seen much increase in prediction accuracy. And also when we add all the genomic features together and further differentiating them between attack or non, well, there was no much increase. So based on this, there was not an impact on prediction accuracy. And we also noticed that there was no changes in dispersion. So the slope remained the one of 50K scenario. Now, into the results from the Canadian colleagues, they actually found much better results, so to say. Here you see the same layout of the slide. And you can notice that for QTL, as soon as they were added to the 50K, they got an increase of accuracy of 24%, which is quite noticeable. And already using QTL alone, which were about 5,000, you have an increase in accuracy of 12% relative to the 50K. EQTL per se didn't help, as we saw in the Netherlands. And furthermore, when we use multi-genomic features, so QTL, EQTL, and differentiated them between attack or non-attack intervals. That also improve a bit the prediction accuracy. And also, they follow the pattern for dispersion was quite similar. So QTL also helped to improve a bit the regression slope. So this brings me to the conclusions. I think that so far we have noticed that including genomic features could increase genomic prediction for dry matter intake, but the results may vary across breeds and datasets. And what we think is the pattern that the Canadian colleague observed. We think it could be explained by the fact that in previous work package, the meta-G was found significant strong peaks for feed efficiency for dry matter intake on the beef cattle, while we didn't find such strong peaks on dairy cattle. So now we have to dig into these SNP effects that were computed and try to see if indeed these are the SNPs that have the strongest effects on the accuracy. And if that is the case, then this would suggest that we really need to find the causal variance, and well, that's not easy because we have to deal with complex traits. Another conclusion is that we haven't seen much advantage of using two-mixture Bayesian approach so far on top of SNP-lab, but I do believe that we have to do a bit extra there, as Bayesian approach could actually improve the weighting of the SNPs. So next, we will focus on specific traits or tissues. As I say, right now, we just use everything. So we would like to focus on specific ones just to see if we may remove some noise. And also, we would like to move to use a Bayesian LV approach, which is developed by the colleagues in Aarhus University. And there, you can provide more detailed information on the annotation layers. So not only at 0, 1 as presence, absence, but you can also tell basically how strong is the association using a continuous variable such as p-values, t-values, or what you have from other meta-GWAS studies. And a part of the project, we also have to do a multi-breed or a cross-breed combined analysis between the Netherlands and Canada. Finally, I would like to acknowledge the BovReg project, the EuroFAANG consortium, Dr. Emre, Karaman, Lukians for the help with the software, Gabriel Costa for the help with AttackSec, and the BovReg consortium. These are the partners. Thank you.